Unit 3, Nutrition and Biochemistry. Make sure you click the link to watch the biochemistry video. So by the end of this unit, you should be able to compare and contrast the four main types of organic compounds. Remember, organic means living. Organic compounds are derived from living things and contain carbon. It must have carbon and hydrogen to be organic. Many scientists can't agree on whether it has to have just carbon or has to have carbon and hydrogen. So for the purpose of this class, I want you to remember that it must have carbon and hydrogen. Inorganic compounds are derived from non-living things, such as water, carbon dioxide. We also call these abiotic. They are non-living. Carbon forms single, double, or triple bonds. Carbon compounds easily form four covalent bonds to create chains, rings, or branches, but you'll learn more about that in chemistry. When a large compound is produced from smaller compounds, as a smaller compound is joined together, this is called polymerization. So when monomers make polymers, again, the polymer is the large compound, and it's made from smaller compounds, monomer. Mono means one, so as one and one and one come together, we get a poly. A macromolecule is a large polymer. A condensation reaction means to make or join compounds. It's also known as dehydration synthesis. When you think of the word dehydrate, you think of removing water or without water. And that's exactly what happens in this chemical reaction. The water is removed to make the bonds. So if you'll notice, the O and the H are squeezed out on the left, and over on the right at the very end you'll see the HOH, that's water, H2O. So dehydration synthesis is also known as a condensation reaction. When you think condensate, think like water on the side of your glass of iced tea in the summer. Hydrolysis reaction means to break apart a compound. So hydro meaning water, lysis means to cut. So water is added, so you notice that we have sucrose and water. Water is added with the sucrase, the enzyme, and the water splits apart. So now we have glucose and fructose. So we have a OH on one, the glucose, and we have an H on the fructose. So the water was split. So it was a polymer, now it's two monomers. So hydrolysis means using water to split the reaction. The four major organic compounds that you need to get memorized are carbohydrates, lipids, proteins, and nucleic acids. So before we get too far into the chemistry, let's talk about nutrition. The general rule is everything in moderation. Make sure you click on that link to watch the video. The FDA, which is the U.S. Food and Drug Administration, provides food data or labels on all products intended for human consumption, and that's only been going on since 1990. 55% calories should come from carbohydrates, 15 calories should come from protein, and 30 or less should come from fat. Do you know how to read nutrition labels? If presented both of these plates, both having the same amount of calories, which one would you choose? The one on the left or the one on the right? Can't wait for lab where we'll talk more about nutrition. For now, let's do the review questions. Make sure you click on the video link for review. Organic compounds must contain which two elements? Carbon and hydrogen. True or false? A monomer is formed from two or more polymers that bond together. False. Monomers form polymers. What is the process of removing water to make bonds? That's the condensation reaction or dehydration synthesis. What is the process of adding water to break bonds? That's hydrolysis. True or false? Only proteins are necessary for survival. False. Proteins, carbs, lipids, and nucleic acids are necessary for survival. By the end of these notes, you should be able to examine the structure and function of carbohydrate molecules. Remember, function is what it does. A carbohydrate is composed of carbon, hydrogen, and oxygen, and it's always in a 1:2 to 1 ratio. So look at C6H12O6. There's 6 carbon, which is 1. 6 goes into 12 2 times, so hydrogen 2 to oxygen 6. So a 1 to 2 to 1 ratio. For every 1 carbon, there's 2 hydrogens and 1 oxygen. A monosaccharide is a simple sugar. They all have the same chemical formula, C6H12O6. There's glucose, produced by plants, fructose, found in fruits, and galactose, found in milk. So count the carbons and hydrogens in each one of these compounds. How many do you find? 6 carbons and 12 hydrogens. That's right. But they're 2 different compounds. Isomers are compounds that are different in structure, but not in molecular composition. So they both have 6 carbons and 12 hydrogens. A disaccharide is formed by 2 monosaccharides. They all have the same chemical formula, C12H22O11. To make a sucrose, we take glucose and fructose. To make maltose, we take glucose and glucose. And to make lactose, we take glucose and galactose. You may remember in your high school biology class gluing these monosaccharides together to make disaccharides. A polysaccharide is formed by 3 or more sugars. Starch is storage for plants. Glycogen is storage for animals. Cellulose is in the cell walls of plants. That's where plants store their carbohydrates. So I heard an old lady once tell me that I shouldn't eat something because it had cellulose in it. And I was like, what? What are you talking about? There's cellulose in every plant. That's a good thing. So when I eat spinach, I'm eating cellulose. It's fine. It's a good carbohydrate. So make sure you know cellulose is a good thing. It's in the cell wall of plants. That's where they store their carbohydrates. The next word is chitin. Pronounce it chitin with the K. Don't pronounce the CH or you might get in trouble. So chitin. This is in the cell wall of fungi. So when you're eating a mushroom, you're eating chitin. What makes all these carbohydrates different from each other is the arrangement of the individual atoms. Its structure, that's what makes it different from each other. So they're isomers. They're like cousins of each other. So here we have a glucose and a fructose coming together. 2 monosaccharides being squeezed together. When they get squeezed together, the water is squeezed out. So notice on the reactant side, we have glucose and fructose, and on the product side, we have water. And what was made when glucose and fructose came together? Sucrose. So this is a dehydration synthesis. Make sure you label your reactions in your notes. This one is a dehydration synthesis. So now we have hydrolysis. Notice that water is on the reactant side this time. So we have sucrose, a disaccharide, and we add water to it. The water splits apart the glucose and fructose. So lysis meaning split, hydro meaning water. So the water was added and then it split apart the disaccharide. So now we have 2 monosaccharides. So make sure you label this example in your notes as well. After this set of notes, you should be able to examine the structure and function of lipids, proteins, and nucleic acids, and then compare and contrast the 4 main types of organic compounds. Lipids are also known as fats and oils, which are fatty compounds. They're made of carbon, hydrogen, and oxygen, with a greater number of carbon to hydrogen atoms and a smaller number of oxygen atoms than carbohydrates, and there is no ratio. Some examples of lipids are fats, oils, waxes. These do not dissolve in water. They are nonpolar. And you remember what water is? Water's polar. So these are nonpolar. Nonpolar does not dissolve in polar. So to make a lipid, we take 3 fatty acids and a glycerol. So a glycerol is a 3-carbon alcohol, and it's combined by dehydration synthesis. A fatty acid is a hydrocarbon chain with a carboxyl group at the end. So how do you make a lipid? That's right, a glycerol and 3 fatty acids. I like to say 3 fatty acids and a glycerol. The hydrophilic end is the water-loving end. Philic means love. Phobic means fear. So water-fearing end and a water-loving end. Even though this is labeled a triglyceride, in your notes label it fatty acids. So there's 3 fatty acids where it says triglyceride, and then your glycerol. Even though we try to reduce our lipids, they are necessary for survival. Much of the cell membrane forms by lipids, and it serves as a barrier between the inside and outside of the cell. This is known as the phospholipid bilayer. We also call it a cell membrane. Lipids are an important source of stored energy in your body. Oils are triglycerides that are liquid at room temperature. Fats are fatty acids that are solid at room temperature. Wax is a highly waterproof coating or layer, and a steroid also does not dissolve in water. This is why when you take steroids when you're sick, you usually gain a little weight, because they are in the category of lipids. So in the making of a lipid, water is removed. So notice the 3 fatty acids and a glycerol. They come together, the water is squeezed out to make a lipid. A saturated fatty acid has the maximum number of hydrogen attached to the chain. It's solid, so think saturated, solid. Unsaturated fats do have a double bonded carbon and do not have the maximum number of hydrogen attached to the chain. So they're liquid. So just remember saturated, solid, and you'll get the other one. A protein is an organic compound made of carbon, hydrogen, oxygen, and nitrogen. Amino acids come together and make a protein. We have thousands of unique proteins. The way I remember this is amino acid. Amino has an N in it, and nitrogen. Nitrogen is in proteins. So amino acids come together to make proteins. There are 20 different kinds of amino acids that form proteins. And a protein has 5 distinct groups. There's a central carbon atom, a single hydrogen atom, a carboxyl group, an amine group. So when you see that nitrogen, amine, that means that if it's amino acid, and if an amino acid joins together with an amino acid, we get a protein. And then there's an R group, which means this could be a small chain of CH2 or a long chain of CH2. Make sure you click dipeptide to watch the video. When 2 amino acids bound together covalently by a condensation reaction between the carboxyl group of one amine and the other amine, they form a peptide bond. So an amino acid comes together with another amino acid, and the water is squeezed out, and we get a peptide bond, and peptide bonds make proteins. A polypeptide is a long chain of amino acids. Some examples of proteins are insulin, hemoglobin, and enzymes. The number and arrangement of the amino acids determine the type and function of the protein. Proteins are a major component of most tissues, including muscle, bodily organs, and cells. The 9 amino acids, called the essential amino acids, cannot be made by the human body. They must be eaten on a regular basis for your body to maintain itself. A nucleic acid is a complex organic molecule that stores important information. There's 2 types, DNA and RNA. DNA is deoxyribonucleic acid. It stores essential information for almost all cell activities, including cell division. And when the nitrogenous base pairs come together, they make a hydrogen bond. So hydrogen bonds are formed in DNA. RNA is just ribonucleic acid. It's a different sugar. And it stores and transfers information for the proteins. We'll study more about DNA later. A nucleotide is a monomer that makes up both DNA and RNA. It's made of 3 parts, a phosphate group, a 5-carbon sugar, and a nitrogen base. So I like to think of this as the house, the pool, and the garage. Some people see a house, a pool, and a flag. As long as I see the pool, I know that that's a phosphate group, and when I see the pool, I know that that's a nucleotide. So the 5-carbon sugar kind of looks like a house. At each corner, at each point, 1, 2, 3, 4, 5, there's 5 carbons there. And then you have the pool, the phosphate group, and then you have the flag or the garage, which is a nitrogenous base. What type of molecule is this? Good job. There's only carbon, hydrogen, and oxygen, and it's a 1 to 2 to 1 ratio. So it's a simple sugar, a carbohydrate, a monosaccharide, all the same thing. So what are the building blocks that make up polysaccharides? That's right, monosaccharides, or sugars, or carbohydrates. If the building block is fatty acid, what does that make up? what is the polymer that comes from fatty acids? Good job, that's a lipid. If the polymer is protein, what's the building block? Good job, amino acids. Remember, amino acids come together to make peptide bonds, and that forms a protein. If nucleotides are the building block, what is the larger unit? Good job, that's a nucleic acid, such as DNA or RNA. If the subunit is sugar, or the monosaccharide is the sugar, what is the macromolecule? In other words, what do sugars make up? That's right, they make up carbohydrates or polysaccharides. What about amino acids? Good job, proteins. And nucleotides? Nucleic acids. Nice review. By the end of these notes, you should be able to know that enzymes are proteins that speed up chemical reactions by lowering the activation energy. Enzymes are reusable and specific. Enzymes are affected by such factors as pH and temperature. And you should be able to recognize that the lock and key model is the enzyme-substrate complex. Enzymes are proteins! Exclamation mark, exclamation mark, exclamation mark. They act as catalysts in nearly all metabolic processes. So what's a catalyst? A catalyst is a substance that speeds up the chemical reaction. So anytime you have an enzyme, it speeds up the reaction. They are essential for all cells. A catalyst lowers the activation energy needed for the reaction to occur. It makes it faster. So it's like a group of teenagers standing around, and they're going to fight. But if people are yelling, fight, 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 then they're going to fight faster. An enzyme is made of amino acids strung together in chains. The chains fold in different ways, which produce a 3D shape, and this determines the function of the enzyme. Enzymes are usually named for the particular chemical whose reaction they catalyze, and they end in the suffix ase. So we're going to be investigating peroxidase in class, as well as amylase. These are enzymes, and they act on the substrate, usually named after them. So peroxidase acts on the substrate, peroxide. Enzymes have regions called active sites, which are slots on the specific molecule that allow a specific substrate to recognize and bind together during the reaction. Where an enzyme links with a substrate, it's called the active site, and this forms the enzyme-substrate complex. It's sort of like your key to your house. Your key is specific, and it fits your door lock. And when they're together, when the key's in the lock, that would be the enzyme-substrate complex. The chemical fit between the enzyme and the substrate must be exact. Make sure you label your notes with the substrate, the active site, the enzyme, where they come together, the enzyme-substrate complex. And notice that the substrate is broken apart into two products in this example. The substrate chemically alters and the products are formed. When the reaction is complete, the enzyme is unchanged and their products are of the substrate. The enzyme is now free to catalyze with additional reactions of this particular substrate. Of course, the environment can influence an enzyme. The temperature, the concentration of the enzyme to the substrate, the cell pH, all these things can cause denaturing of an enzyme. Denaturing means to break down. And if an enzyme is denatured, it stops working. So, on the graph on the right, top right, the optimum temperature, the best temperature that this enzyme works, is around 37 degrees Celsius. So if the enzyme is at a different temperature, it's not going to work at its best. And it could be denatured if it gets too hot or too cold. Down at the bottom of the graph, there is a pH graph. So amylase, which is in your saliva in your mouth, it works best at a pH of 7. So if your mouth is too acidic, if there's too much acid in your mouth, that enzyme's not working properly. We're going to investigate this a little bit more in lab. You will see these on your final exam, so make sure you understand the differences in enzymes. Make sure you get this labeled in your notes. This is a hydrolysis reaction, Why? That's right, water was added to split apart the substrate.